So I'm excited to talk to you all today. Thank you for joining me this afternoon. Please just keep your audio muted and your video feed off. It doesn't matter, whatever settings you have now are fine. But I ask that if you have any questions, feel free to email them to me at nperkons@gmail.com, either if you're watching now or watching the recording. I have some time at the end that I'll get back to some of those questions, and the others I'll respond to by email. So to get started, this is a standard T2-weighted proton MR image of a rat with hepatocellular carcinoma. And for those who aren't radiologists in the audience that can't pick it out, the lesion, or the tumor in this case, the hepatocellular carcinoma, is denoted here by this red circle. And what this image represents is really an axial cross-section of a rat liver that bifurcates the tumor, as shown in the schematic over here on the right. And this is an example of a conventional imaging approach that helps us to measure certain things about tumors, like their size, their location, and their vascularity. A goal of my thesis research has been to go beyond some of these metrics to functionally assess tumors and ask a couple of additional questions, like, are tumors viable? What sort of metabolic activity do they have? Do they respond to treatment? And at this juncture, the question, of course, is, well, how do we go about doing that? And luckily, molecular imaging provides a roadmap for answering these kinds of questions by providing a groundwork for the evaluation of disease from the time of diagnosis all the way out to treatment follow-up. And there are several classes of molecular imaging agents, and I'm only going to talk about two today. The most well-known that hopefully many in the audience may have heard of before is positron emission tomography. It uses radioactive nuclei to detect probe accumulation and metabolism. And it's frequently used in cancer, specifically with glucose, or 18-FDG, to look at the localization of cancer cells based on the trapping of this molecule. Another modality that I'm going to talk about a little bit more in detail that most of you probably haven't heard about before is DNP MRSI. And this is an example in which pyruvate is labeled and used to look at liver cancer, or look at prostate cancer rather. And for those in the audience right now, what you can think of this almost as a correlative PET. I'm going to talk about how the two modalities are different, and I'll get to that shortly. But I've given you two examples here of two different types of imaging agents that are used. But another question that I sort of sat with in the beginning was, how many different molecular imaging agents are there out there? And is there really a limit to what we can use? And this is sort of a nuanced answer. There are a lot, but of course there are a few caveats. And this is a map of all of cellular metabolism that is quite frankly overwhelming. All of the nodes, or the little circles in here, are metabolites, and the edges that are connecting them are enzymes. And in molecular imaging, the contrast agents that we use are frequently these nodes, these metabolites, or a derivative of them. But only a small percentage of those in this graphic have ever been used as molecular imaging agents, and a much, much smaller fraction of them have ever been used clinically. My hypothesis going into this was that functional screening could help in the translation of these probes by helping us to find more effective applications for them. And I want to take a second here to just say, I know I keep using the word probe. For those who are getting confused or not sure what I'm meaning, another way to sort of think about this is it's like a contrast agent, at least for the context of this talk. So we're injecting molecules that we can use to look at how the body, or in this case, as I will talk about, cancer specifically, takes in that contrast agent and modifies it in some way that can maybe tell us the importance of that, or the importance of an underlying metabolic pathway. So this was my hypothesis going into it, and I'll walk you through the approach I used over the last couple of years. So the first step of this process was to identify a list of candidate probes for a specific modality that I wanted to screen. And as I promised, I'll take a little bit more time to talk in detail about dynamic nuclear polarization. It's a technique that helps us to enhance the signal available on stable carbon isotopes. And well, what does that mean? So there are metabolites out there like pyruvate, this is an example, in which if you have it labeled with a C13, which is not radioactive, you can excite that nucleus so that with imaging techniques like NMR or MRI, we can get more signal out of it. And so the way that happens is with a machine called a hyperpolarizer, in which you mix this metabolite with a free radical, you place it in the hyperpolarizer, it brings it down to about absolute zero, you microwave it in a magnetic field, and then you rapidly shoot it out, bring it to room temperature, and you inject it into a subject of interest. And this graphic that I've made, it's showing us injecting it into a rat, but it just as easily could be injected into a human. And one of the key points I want to make right here is that this whole setup has been optimized to shorten the amount of time it takes from the time that this agent is prepared to the time it's injected. And that's because while the agent isn't radioactive, the excited state that it's in, which I'm not going to get into the physics necessarily behind it, but save it to say that that is a short-lived state, and we want to optimize the amount of signal that we have because we lose it with time. So shortening the time to imaging really helps there. But so what you need to know now is we've injected this molecule that has a label where the yellow is on the carbon. And when the body sees that, it treats it like just any other metabolite, like pyruvate. And it can convert it in sort of a simplified fashion to other metabolites, like lactate or alanine. And this process is mediated by enzymes like LDH or ALT. And this, of course, as I mentioned, is a simplified diagram of the fates of pyruvate. There's obviously many more for the metabolists or those who specialize in cancer metabolism out there. But these are three fates that we can readily see with this technique. And using NMR and based on chemical shift, we can measure this interconversion between these different states. So for people that haven't seen this, this is an example of several spectra that are collected in time. And each of these peaks corresponds to different metabolites. So this really tall one is pyruvate, the nucleus we injected. The smaller one over here is lactate, and this one over here is alanine. And in time, we could see a small evolution between these different states that helps us to say something about how active these enzymes are within a tumor. And this data is actually coming from a rat with hepatocellular carcinoma. And so how does this relate to imaging? So back to that first proton image that I showed you, if you were to inject pyruvate that's been labeled in the way I mentioned, you could superimpose a second image on top of that first one. And with this second image, we could see inside each of these little boxes, which radiologists will call voxels, you can see that little trace that I had shown you before, a little spectrum in each one. And what that helps us to do is to evaluate metabolism locally, including within the tumor, which I kept the red circle around. And for each of the peaks within this, we can see, for example, pyruvate in red now, or lactate, or alanine. And by collecting a bunch of images in series of this type, we could say something about the metabolism of the tumor and also about the enzymes that are present there. And so that is where this diagram that I showed you earlier, that comes from the sum of all of the signals in here collected at multiple points in time as the rat is breathing. So hopefully this provides an overview of how this imaging technique works and gives a foundation for the next steps of the project. So back to this crazy diagram. So the question I had at this point was, well, what imaging agents exist in here that other people have used? And I started by looking at the literature specifically for the technique I'm talking about with DNP. And those are signified by the purple nodes on the graph. These are imaging agents that other labs, including ours, have used before. And when they've injected them in either in vitro or in vivo systems, these investigators have been able to measure the accumulation of a bunch of intermediates, which are the pink nodes. And what's powerful about this technique is the relative transition between the purple and pink nodes tells you about the enzymes that are connecting them, which I've represented with these dashed black lines. So what I'm trying to get at here is that with this technique, we could look at the activity of different enzymes based on the relative interconversion of the purple and pink dots. So with a list of candidate probes in mind as a jumping off point, the next step was to screen for a specific application. And I focused on cancer. And so to do this, I turned to CRISPR selection screening. And in this example, there's a bunch of cancer cells that are situated in a plate. And normally these cancer cells would grow exponentially and take up the entire plate. And you'd expect, because there's equal amounts of these different colored ones, that at some time later look sort of like a pizza pie where they each take up an equal slice. But what I haven't told you yet is that each color has had one enzyme removed. And so by removing one enzyme from otherwise equal cancer cells, we can look at some time later and learn about what the importance of those enzymes we took away are. So if we look sometime later and see how the distribution of cells has changed, we see that, for example, these gold cells, they were more likely to proliferate without a particular enzyme. So that the loss of that enzyme gave them a growth advantage. Whereas with the blue cells, those cells, when they lost whatever enzyme they lost, they didn't do as well. They had a relative growth disadvantage. And this is what I was going after in cancer in particular, because I wanted to know what cells behave like the blue ones, which enzymes that when we take them away from a cancer cell make them grow slower. So this in total is called negative selection screening, and this helps us to identify enzymes that are important for cancer growth. And as a reminder, I mentioned that DNP MRSI probes can help us to look at enzyme activity. So that's how this is going to link back to this CRISPR screen. So when we inject something like pyruvate, we can get at the activity of enzymes like LDH and ALT based on the formation of these other metabolites. As a preview of sort of what's to come, I'm going to tell you that hepatocellular carcinoma cells that are lacking this enzyme, LDH, they behave like the blue cells. So when they lose this enzyme, LDH, they're less likely to grow. And that tells us a few important things. It tells us, well, one, they need this enzyme to grow, or without it, they don't grow as well. And the second, that this enzyme is pretty important. So perhaps imaging with pyruvate and looking at the relative formation of lactate might tell us something about that tumor and might tell us whether or not a treatment strategy looking directly there is working. But first, a more global approach. So I used two parallel approaches to look at imaging targets very broadly. The first was a genome-wide cancer screen. And for this, I turned to some publicly available data sets that were made by the Broad Institute. And they cover over 18,000 genes, which includes basically all of the edges in this graph, all of the enzymes. And they were in 625 cancer lines, and 20 of those are hepatocellular carcinoma. So what this was used for was to just get a broad picture of what genes and what enzymes are important in a bunch of different cancers. And from there, I took on a second approach, which was to develop my own CRISPR screen that was a little bit smaller and focused just on those dashed black lines in here, just the enzymes that were responsible for probe metabolism, so I could ask more specific questions with a higher specificity. And without getting into too much of the design there were more replicates and more controls that gave me more statistical power. So with this as a starting off point, using that first approach, the genome-wide approach, I was able to identify in 17 different cancer types an imaging agent that might work well. And the way that worked was I was able to identify a dashed black line that corresponded to an imaging agent in each of these cancers that was more important than some others. This was a lot of data, so the next step was to just sort of focus in on one particular cancer, a compelling example that I wanted to learn a little bit more about and test out whether this could work as an imaging approach. And as I've alluded to a few times already, I decided to focus on liver cancer, and in particular, the most common form of liver cancer, which is hepatocellular carcinoma. And hepatocellular carcinoma, it's a leading cause of cancer death, and it typically develops in the setting of advanced irreversible injury. It's very prevalent in Asia and West Africa due to hepatitis B virus. It's the fastest rising cause of cancer death here in the United States, mostly because of the increasing prevalence of non-alcoholic fatty liver disease. And unfortunately, systemic therapies have only provided modest increases to overall survival. They've only extended survival by about 3 months at this point. So with that general screening approach, by focusing in on hepatocellular carcinoma, I was able to ask the question, what genes and what enzymes are more important in this cancer than any other cancer? And what I found was that among these, lactate dehydrogenase A, the most common isoform of LDH, that protein I keep talking about, was more important as an enzyme in hepatocellular carcinoma as compared to all other cancers. This is a way to graphically show that. LDH is known in the cancer field to be a very important enzyme generally. But what this data shows is that in liver cancer, and in particular HCC, there's this leftward shift, which signifies that this enzyme was more important to the growth of HCC than it was to the growth of any other cancer. Also, an interesting finding that fell out of this data set was FGFR1 was a significantly dependent hit in this group. And that's interesting because FGFR1 is one of the targets of sorafenib, one of the few therapies that we have for HCC. And so at this point, I then went to the second approach, which was to validate these findings. And with that imaging-focused smaller library, I was able to confirm that LDH was a hit in one of the cell lines included in that broad screen, the SNU449s. And then I actually performed this screen in a cell line derived directly from a patient here at the University of Pennsylvania. So a patient at the hospital at the University of Pennsylvania had a biopsy performed, and that biopsy was injected into an immunocompromised mouse by colleagues in our lab. And that grew into a tumor, which expanded the cells, and we were able to perform the exact same screen on those cells and found again that LDH was among the significant hits in this cancer. And what does that tell us? Altogether, the point here is that LDH is a very important enzyme in liver cancer. It's sort of known that it's important in many cancers, but it's more so in liver cancer than some others, and that has some implications for imaging. So based on the fact that this is important and that we can measure these different fates with the technique I keep talking about, it suggests that if we were to inject pyruvate, we could say something about the activity of LDH in tumors based on the formation of lactate. And just as a reminder, the way this would work in an imaging situation would be by collecting such spectroscopic images. Another finding that comes out of this is that the CRISPR screens told us that if we genetically remove LDH from cancer cells, they're less likely to grow. So that suggests that LDH inhibition is a therapeutic strategy to treat HCC. So if we remove LDH genetically, they grow less. It makes sense that if we were to inhibit it with a drug or a small molecule that specifically binds to this, we would be able to slow the growth of HCC, a cancer that doesn't have too many approved therapies at the moment. Another powerful piece of this picture in terms of the imaging technique that we're using is that if we were to effectively block this step, we can measure the efficacy of that based on how much of this is formed when we inject pyruvate. So my hypothesis going into this is if we use an LDH inhibitor, we would prevent the flow of pyruvate to lactate, so this peak here would get smaller. And so I'll show some images. So this is what a baseline image just focusing on the tumor looks like. This shows us the ratio of lactate to pyruvate, which is a standard metric in the field for measuring flux through the enzyme lactate dehydrogenase. And we see that there's, according to this scale bar, an accumulation of signal within the tumor, signifying that that enzyme is active. And this data is, again, just a visual representation of some spectroscopic data where there's a lactate peak here, a pyruvate peak here, and there's multiple peaks collected in time. At this point, I give an LDH inhibitor, something that blocks that step that converts pyruvate to lactate, and do the same experiment, essentially. And what we found is a profound reduction in the amount of lactate produced from pyruvate. And we see that here because the color is essentially not there anymore. The tumor isn't producing any lactate. And it's even more profound when you look at the spectroscopic images. When you see that there's not that peak here corresponding to the red bar, the lactate isn't being generated anymore. So the next piece of this puzzle was to validate this on a larger scale. And so in order to bring together multiple animals that were studied, we use some of the techniques that are standard in the field, which is to take the area under the curve of these dynamic signals by integrating pyruvate, lactate, and alanine amounts and comparing them across multiple experiments. And that helps us to generate graphics that look like this, which are a bit unwieldy, so I'll just focus on a few key examples. So one of the key findings here is that lactate to pyruvate. So this is telling us the amount of lactate relative to pyruvate before and after giving an LDH inhibitor. And we see that there's a marked reduction. So what this blue box is telling us is that this step is effectively working. We're not seeing this going here. Now, because this hasn't gone here, and I admit that this is, again, a very reductionist view of the fates of pyruvate, but these are just the ones we're imaging at the moment. There's more of this that accumulates, which we see over here on the left, and a little bit more that's accumulating here pops up over here. It's shunted a little bit towards alanine, which is shown in this blue box over here. So at this point, the next question was, of course, well, what's the effect on tumor growth? Because we saw in the in vitro setting that LDH inhibition slowed tumor growth. And we, in fact, found that that was also true in this rat model of liver cancer, where the tumor growth rate before versus after giving the drug, it increased the doubling time, which means that the tumor growth rate slowed. So the amount of time it took for the tumor to get twice as large before drug was significantly smaller than the amount of time it took afterwards. And we found that this relationship was dose dependent. So the fold change in this doubling time represents this point divided by this point. And we found that it was much larger for a larger dose of 20 milligrams per kilogram as compared to 10 milligrams per kilogram. And then next, the most exciting and sort of clinically relevant piece of all of this was the prediction of efficacy. And so when LDH was inhibited, I found that post-inhibition alanine levels correlated with the degree of growth slowing. And what that means is, so again, we blocked this step and we ultimately saw a little bit more of flux or transitioning of pyruvate going this way. And we can see that to some degree in these example images that I keep showing of alanine production. And using a statistical model that takes into account all of these factors and the amounts of the metabolites, we found that alanine was significantly predictive of growth slowing. So what does that mean? What that means is based on how much alanine we saw after giving the drug, we could predict whether it would take 3 or 4 or 5 days for the tumor to double to some degree of certainty. And why that's exciting is that this is all happening at the time that we give the drug. So you can imagine a clinical scenario in which you give the drug, repeat the imaging, and you can titrate your dose based upon the desired effect. And so this brings us to the end of the first part of this presentation. So I just want to summarize some key points. The first was that CRISPR-based screening helped me to isolate imageable dependencies in 17 cancer types. And LDH was found to be a differential dependency of HCC. So HCC was found to depend on LDH for growth much more than the other cancers. DNP pyruvate helped to facilitate local quantification of that inhibition. And this precipitated a dose-dependent growth slowing in the cancer. And ultimately, alanine formation after giving the inhibitor predicted the degree of growth slowing at the time that we gave the drug. So the next piece of this project was to address concerns for translation. So these are going to be a little bit more technical details that I will get into shortly. And one of the things that I would say is an important note that motivates a lot of these questions is that the concentration that we're using of pyruvate is up to 1,000 times the equilibrium serum concentration, which definitely is a cause for concern, and I'll explain why I don't think that is necessarily so among some folks in the cancer metabolism world, and especially in the pet world. So I will go through how I address some of these concerns and what that means for eventual translation, hopefully. So the first question is, what are the pharmacokinetics of the injected species? So we're injecting pyruvate, but in order to get that an excited signal, we use a free radical to help excite it. So what's happening to those? How is equilibrium restored, and how quickly? So to do this, I essentially gave pyruvate the same way I always do, except I just didn't put it in the hyperpolarizer. I still mixed it with the free radical, and then measured the serum serially. And what I found was that while shortly after giving an injection of pyruvate, serum pyruvate levels spiked, they pretty rapidly normalized. And within 30 to 45 minutes, they were back to close to their baseline levels, which of course begs the question, what in the world happened to all of that pyruvate? So we know for a fact that some of that pyruvate is being metabolized in tumor cells, for example, in the liver and other regions of the body. But we also had the hypothesis that perhaps the kidney was clearing some of it. And so without getting into too much kidney biology, how it works is that your blood is filtered, and a lot of metabolites within that filtrate have to be reabsorbed in order to not be excreted into the urine. So what that means is that we collected urine before and after giving these injections and monitored for pyruvate amounts. And I found that there was an increase in the amount of pyruvate in the urine afterwards. And what's also not shown here is that there was a rapid increase in the amount or the volume of urine afterwards. So the total amount of pyruvate in urine afterwards was pretty high. So in answer, like what happens to the pyruvate? Some of it's metabolized, some of it's cleared, but we definitely see a substantial amount of clearance in the urine. And because we had all of this urine stored up, we had it in the fridge, and one of the research techs and I was looking at it, and I was like, well, this is interesting. So this is a sample before giving the pyruvate, and this is a sample after. And clearly it should not be purple. So we were starting to think about reasons that that could be the case. And in talking with members of some other labs and doing some reading, we started to hypothesize that this could be the free radical reacting with light and some of the proteins. So, and I confirmed that eventually with EPR, and we did indeed see that there was radical accumulating in the urine. And I should note here that this isn't a concern necessarily for human clinical translation. There's a different radical that's being used at some of the sites that are doing clinical trials at the moment that they're able to precipitate out. But this is of interest for animal experiments, and in the case that in a clinical human experiment, it isn't able to be precipitated, this is a place to potentially look for it in the urine. The next question was, are DNP-based metabolism quantifications reproducible? So the easy way that I did this experiment was 45 minutes apart. I did the same experiments and looked at all of those metrics that I've been showing on the past slides, and I didn't see any statistically significant differences in 9 rats between the quantifications. Another is, do these quantifications of metabolism correlate with other measures of enzyme activity? And for this, I returned to a similar experiment to the ones I've talked about before, where I used an LDH inhibitor, except I just varied the dose between a couple of different levels. and I measured the same sort of measurements, so lactate to pyruvate before and after giving a drug. And then immediately after this measurement, I sacrificed the animals and upon necropsy harvested the tumors and homogenized them and used a spectrophotometric assay of enzyme activity and correlated that with the imaging measurements. And what I found was that DNP-based quantification of LDH activity correlated significantly with those measurements. Another question was, does probe concentration affect the DNP-based measurements of metabolism? And it helps in this case to motivate this question to talk about the setup a little bit more again. So there's a hyperpolarizer that prepares the substrate and it's rapidly injected into a subject, which in all of these experiments I talk about are in rats. And so when it arrives from the hyperpolarizer, it goes into the tail vein and goes through the circulation of the animal, gets to the lungs, goes back to the heart. And because it's hepatocellular carcinoma, it reaches the tumor through the hepatic artery. Will be a little bit more important later in the talk, but it's important to note that liver cancer, hepatocellular carcinoma, is supplied by an artery, whereas the liver itself is mostly supplied by a venous system. It's draining most of your gut. And so once it arrives there, it goes through vascular beds. And one of my hypotheses about what might happen with different probe concentrations was that because we're over 1,000-fold the normal serum concentration of these metabolites like pyruvate, that this step of going from the capillary beds into the tumor cells and specifically diffusing into the tumor cells through something transporter known as MCT1 would become rate limiting. So if we were to just increase the probe concentration, there'd be a disproportionate increase here of just the amount of pyruvate that's in the vascular bed as compared to what's in the cells where the metabolites are being formed. And I was able to measure that by doing 2 injections at slightly different concentrations at 80 millimolar, which was my standard concentration I used for all my experiments in this thesis, and 160. And what I found was that the relative amounts of the metabolites to pyruvate itself went down. So lactate to pyruvate goes down because there's more lactate. Alanine to pyruvate went down as well, though not statistically significant here. And the fraction of pyruvate signal total went up. And then one of the more fun questions was, how can we reduce experimental complexity and save time? And this, again, just to return to an earlier point I made, time is signal in all of these experiments. So the faster you can get the injection to occur, the better. And so back to this drawing, the original setup we had back when I started my PhD, it would take about 4 hours to get one of these experiments done. And so we would have maybe 5, as Dr. Gate is laughing behind me. So we would prepare the sample, and we would actually take in a syringe and run it, because these 2 devices are actually in different rooms. We would run it through a hallway and then inject it, and this would take about 3 people. It was very experimentally complex. About halfway through my PhD, we introduced this pump here that automated part of it and reduced it down from 3 people to 2 people. And so I can't have a talk on DNP without some videos. So this is a video showing one of the middle ground setups. So there's a pump here, the pyruvate's being prepared. And at this point in time, I have to yell across the room to synchronize all of our experiments. And Omar, who's a lab technician with us at the time, would help to synchronize the delivery of the pyruvate into the magnet. And this is connected directly to the rat's tail vein. And then at a certain point in the timing of all of this, I would run over. So you can see that there's only 2 people required here. I would run over and start the imaging sequence. But of course, this might not be super reproducible. It's definitely better than what we had before. So a more recent innovation was using an Arduino to help program all of this. And so the Arduino here can control the start and finish of the scans, timing them so that it's more reproducible. And a key part of this actually is that it only requires 1 person to do all these experiments, which has been a significant development for the lab. At a very certain prescribed time, I start the sequence and it goes ahead. And so this is a process that I, in particular, am continuing to sort of develop further, and it's been a fun pet project in the lab. And so another thing that I did to help with sort of the translation and reproducibility of all of this is I developed a MATLAB pipeline. I'm not going to get too much into detail, but I did this in collaboration with some folks in Dr. Rahim Rizzi's lab that helps me to automate peak picking and to just do all of the spectral processing in near real time as it comes off the scanner. And so that brings me to the end of this pipeline. So I want to take a few minutes now to discuss some offshoots of this project, some next steps, and some of the data that we have to support those next steps. So the most obvious next step would be to validate this model in additional cancer types. So I mentioned already that we focused on liver cancer, but there's 16 other cancer types that I haven't talked about, and that is an active area of investigation in the lab. Another would be to leverage screening results to guide the development of new imaging probes. I sort of threw it out there that many of these metabolites out here could be used as potential imaging agents in the future. Well, the screening approach, number one, the DepMap whole genome screening approach, helped point to some potential imaging probes that we could use for a few new cancers. There's 4 new ones that weren't included in the original 17 that are specified by these yellow dots here on the graphs. So that's another potential to look at developing new agents. And I'm working on this in collaboration with Dr. Steve Kadlacek and Dr. Joe Wildenberg with a molecular dynamics simulation that helps us to pick out which among those probes are the best. And unfortunately, I know that this is not going to play well in the live stream, but these 2 videos here will show you a simulation of what pyruvate looks like as it moves through water. And this helps us to model a key parameter, the T1 relaxation time, which governs how long we can get signal out of a nucleus. And I keep mentioning that time is signal. Basically, if a molecule has a larger one of these numbers, a larger T1 relaxation time, it's a better candidate, and it's better for us to invest more time in that one. So this is what it looks like in 1 femtosecond, a little pyruvate molecule dancing around in a cage of water. And this is what it looks like in 1 picosecond. So some cool animations about other things that are going on in the lab that I've been involved with. Another next step would be to utilize an analogous screening approach to screen for changes in cell state. So this is actually returning to that second modality of screening I was talking about, that smaller imaging focused screen. One of the things that I have already done was to look at the impact of growth media components on cancer cells' ability to grow. And so here, what this experiment is meant to highlight is I have complete media, which has a lot of nutrients, a lot of oxygen, and a lot of sugar, compared to media that has less of that. So cancer cells that have a lot of nutrients up here or not so many nutrients. And I wanted to see, well, could we use imaging to pick out the areas that have less nutrients that are still alive? And where this is clinically relevant is for potentially if a tumor has outgrown its blood supply, or someone like an interventional radiologist has performed a procedure that has artificially blocked a tumor's blood supply, but the cells have remained viable. And with the screening approach, I found that several NADPH dependent reductases of the thyroidoxin system help to distinguish this group of cells versus this group of cells. And these are some of the enzyme hits that I had, and this is just highlighting that many enzymes in this pathway overall were very significantly more important in conditions where there was nutrient limitation as compared to when there was nutrient abundance. And to bring it back to imaging, we've already started to do some pilot experiments where we use a different DMP probe, dehydroascorbic acid, and we look at its conversion to ascorbic acid, which is mediated by those very enzymes that I said are more important under conditions of nutrient restriction. And so this is stacked spectra of DHA as it's converted to ascorbic acid, which you can see in these 2 peaks here in a rat liver in a tumor as well. Another approach that would be useful that we have started to work on is to utilize this same screening approach to look at other types of imaging probes. I've talked about DNP and MRS probes sort of ad nauseum here, but there's of course many other imaging approaches, many other molecular imaging approaches, and the first of course that jumps to mind is PET. So one of the first steps I've taken in this direction is to look at the relative importance of hexokinase, which is the enzyme that's responsible for giving you a PET signal, essentially. So when 18F-FDG is taken up by a cancer cell, it gets metabolized by hexokinase, and usually hexokinase 2, it gets phosphorylated. and it gets trapped within a cancer cell. And that's what causes the accumulation of that, causes a signal sort of indirectly. And so I looked at what the implications were for hexokinase 2 in the context of that broad depth map screen that included many cancer types. And I found that a lot of cancers depend on hexokinase 2, but multiple myeloma, interestingly enough, was differentially dependent on hexokinase 2. So what that means is many cancers needed hexokinase 2 to survive, but multiple myeloma actually stood out as one that was statistically significant in that it required it more than many other cancers. And why do I keep talking about multiple myeloma? Well, this approach is showing that, and it's consistent with clinical data where FDG PET is a preferred modality for multiple myeloma in particular. And this includes for functional assessment of response to treatment. So this is tying the HK2 finding in multiple myeloma to a clinical phenotype. And in xenografts, HK2 knockout impaired FDG uptake without affecting size. And in human patients, it was found that multiple myeloma was not FDG PET avid, had a lower expression of HK2. So just sort of bringing together this whole approach that I've used in a different imaging modality with just some preliminary data that also sort of serves almost even as a motivation for the work that I did, in that it suggests that maybe we're not too far off in getting DNP closer to the clinic. And of course, completing the automation of the fluid handling and sequence timing so that we're saving as much time as possible with the Arduino setup as well. And so as one other next step that isn't quite a next step because it already happened, just sort of a logical offshoot of all of this work, was to use DNP pyruvate not as a tool for looking at systemic therapy, but looking at as a tool for looking at local regional therapies. And the data that I'm going to briefly talk about here is from a paper that we just published in hepatology recently. And what this paper covers is the case of a procedure that's frequently performed by interventional radiologists. And so to treat liver cancer, interventional radiologists will sometimes go through the blood supply with a catheter and occlude the blood supply to the tumor, which in this case is an artery as compared to the rest of the liver, which is mostly venous. And it uses small beads to block off the blood supply. And what we showed using DNP pyruvate, the molecule that I keep talking about, was that the ischemia, or the oxygen and nutrient loss that happens after you block that blood supply, leads to changes in the tumor that precipitate metabolic reprogramming. And without getting too much into the nitty-gritty, the DNP pyruvate, that injection, was more sensitive to viability than conventional imaging was. It confirmed this metabolic reprogramming based on some of the ratios I've talked about, and it predicted tumor viability on H&E. So it predicted a histopathologic diagnosis. And so with that, there are many grants that have made this possible, a few that have been awarded to me directly that I'm greatly appreciative, from the National Cancer Institute and from the Society of Interventional Radiology Foundation. As my mentor previously alluded, this has led to a number of papers in my time here, and there's a few that are under review and in preparation. The work that I talked about today is actually largely unpublished, as specified by the blue arrows, although a few of them are under review, and we're hoping to hear back shortly. A project that I spent actually the first year and a half of my time in the lab working with Dr. Elliot Stein and now Gabriella Pilla on was the development of a biomedical device for the treatment of liver cancer that we have actually just submitted an STTR on and are hoping to hear back soon. There have also been a few collaborations I've done in the lab that are signified by the purple arrows, using some of the tools and techniques I learned, and a few collaborations outside of the lab. And so with that, I would like to thank my families, to my dad, my mom, my sister, Bob and Nancy, my grandparents, and of course, the person that's in all of these photos that I haven't said by name yet, my fiance, Michelle. Thank you for all of your love and support for the last couple of years. And as I am about to return to my medical phase of my training, to my many friends from medical school, very broadly, can't take time to name everyone, but also the MD PhD program down here on the left, my learning team from the medical school over here on the right, friends from graduate school, from collaborating labs, from the rowing team that has been a large part of my experience here at Penn, friends from back in undergrad, and from Jefferson's Med School. And of course, thank you to the members of the Penn Image Guided Interventions Lab, many who are shown here, for all of your help throughout the years. And of course, now I'm going to feel bad for posting this, but no. my biggest cheerleader, Terrence Gade, I was expecting that I was going to get roasted in my introduction, so now I feel bad. And of course, thank you, of course, to the many members of the GATE Lab, to the RISI Lab, Stephen and Greg in particular, Dr. Simon, my thesis committee members, many who are here with me now, Dr. Schnall, Dr. Chodosh, Dr. Mankoff, Dr. Chakas, the Penn Interventional Radiology section for supporting our lab in particular, and me as well as I develop my skills here, the PET Center, Small Animal Imaging Facility, Dr. Tony Mancuso, the graduate group, and of course the medical scientist training program that has helped to integrate my medical degree and my graduate degree while I've been here. And so with that, I will open up to questions. So feel free to email them in to my email here at nperkons@gmail.com, and I will pull some up on my other laptop during the public part of this presentation.